quien es profesor asistente en el Ryan Stephanie Lane Computational Biology Department and the Carnegie Mellon University. Dr. Carja obtuvo su grado de licenciatura en matemáticas en la Universidad de Chiusa en Rumania en el 2008. Después de sus estudios de licenciatura, se enroló en el programa de biología de doctorado de la Universidad de Stanford, la cual concluyó en el 2014 bajo la dirección del doctor Marcus Feldman, estudiando evolución de la genética y epigenética y diversidad en medios ambientes cambiantes. Su primer postdoctorado lo realizó en la Universidad de Pensilvania, el cual concluyó en el 2017, para continuar con otro postdoctorado ahora en la Universidad de Stanford y en el Chan Zuckerberg BioHub, que concluyó en el 2019, y a partir del 2019 es profesor asistente en la Carnegie Mellon University. La doctora Carja ha recibido grants del NIH y de la Fundación Bilateral Estados Unidos-Israel para conducir su investigación. Actualmente en su grupo joven de investigación tiene bajo su dirección a 6 estudiantes de doctorado. Participa activamente en labores docentes en la universidad. Es profesora de los programas de Biological Modeling and Simulation y de Essential Mathematics and Statistics for Scientists. Y de los diferentes premios que ha recibido, me gustaría resaltar uno que ella menciona, que desde pequeña en Rumania ganó diferentes premios en competiciones de matemáticas, y bueno, eso queda claramente reflejado su amor por las matemáticas en lo que realiza como tema de investigación. El laboratorio de la doctora Carja se enfoca en comprender cuantitativamente la arquitectura evolutiva de sistemas inteligentes y colectivos. Utiliza herramientas de sistemas dinámicos, teoría de redes, genética de poblaciones y aprendizaje automático para explorar procesos biológicos complejos. Su trabajo destaca la importancia de la estructura espacial en las dinámicas evolutivas, y su investigación tiene como objetivo proporcionar conocimiento sobre cómo la estructura de las poblaciones influyen en la tasa evolutiva en las variaciones genéticas. En estos temas, la doctora Carja ha publicado alrededor de 25 artículos en revistas de alto impacto, y hoy nos va a hablar precisamente acerca de una teoría evolutiva de la dinámica en poblaciones, en la estructura de poblaciones complejas. Oana, thank you very much for accepting the invitation and for the class yesterday with the students and for the seminar today. Please go ahead.
Thank you so much, and thank you. I think I understood most of it. Thank you so much for the invitation. I'm so sorry I cannot be there, and hopefully I get another opportunity sometime soon. So today I wanted to share some of the work that we have been doing in my group since we started. We essentially started January 2020 in the pandemic. And we've been slowly building up the group, and am excited to share some of our results. So in my group, at a very big picture level, we study the shape and structure of complex biological systems, from the arrangement of cells in tissues to the arrangements of individuals in a population. And I'll explain more what I mean by that. This idea of shape and structure is, and us trying to understand the shape and structure that we see around us, is by no means new. And actually, one of my favorite books is a book that is more than 100 years old. It was written by Darcy Thompson in 1917. And it's called On Growth and Form. And in this book, we see some of these first images of the shapes of cells or other organisms that allow us to sort of dream about the biological world around us. And another reason why I particularly wanted to mention this book is because there was a recent article by one of my favorite science writers, Philip Ball, where he kind of rivals this book with The Origin of Species. And he claims, hey, this is another more than 100-year-old book that still to this day shapes science around us. And I particularly wanted to focus on this part of the article. I think the whole article is just really interesting and beautiful to read. But I particularly wanted to focus on this part because it's relevant to what I'm going to tell you about today. So Philip Ball says, behind it all is the message that shape, form, pattern in the natural world is not arbitrarily sculpted by Darwinian evolution for adaptive supremacy, but follows deeper mathematical laws running through the living and non-living worlds alike. Thomson sometimes implied that this view was an alternative to Darwinism. And he continues, my instinct is that natural selection wouldn't be sensitive to such fine distinctions. Here, at least, I'm with Thomson, right? So this begs the question, and particularly for me, I started out in mathematics, but I started becoming fascinated by evolution and evolutionary biology. So for me as an evolutionary biologist, it begs the question, does a system structure, right, its shape, its structure, its population structure, serve an adaptive function, right? Or is the pattern, as Darcy Thompson and Philip Ball would have argued, just an incidental consequence of growth? Does topology play no evolutionary role, right? So that's the question that fascinated me and is part of the research program of my lab. And there's starting to be hints that indeed structure may matter, right? So for example, this is a paper that came out very recently that poses this very interesting hypothesis that the shape of the lung, right? We know that the lung looks kind of like a tree. It has this fractal pattern. And it's been claimed that the dispersal of the influenza viral populations within these tracts, within these lung tracts, actually shapes their evolutionary potential. So the shape of the lung, the structure, this fractal structure, could potentially affect viral populations that are constrained by these structures. We see these types of tree-like structures in many other biological systems, not least in different types of cancers. For example, there are particular cancers called ductal carcinomas that grow along and are restricted by the shape of the ducts. And again, we don't really know how these spatial constraints potentially affect their evolution. So this opens the door to some of these questions. Does the tree-shaped structure of the lung affect evolutionary dynamics for bacterial or viral populations? For example, we're working with collaborators at University of Washington looking at cystic fibrosis patients. Do spatial constraints formed by ductal structure shape patient outcome, evolutionary dynamics in ductal carcinomas or other types of cancers that are also fast evolving ecosystems? Or let's say we think of another question. Imagine a pathogen spreading through a host population, right? We are starting to see and to get more data and to have access to the topologies that viruses are evolving on, right? As they spread between different hosts on the social networks in the world. And indeed, different pathogens potentially have different host social networks that they're spreading on. There are these beautiful papers looking at the social networks for viruses, but because bacteria have different types of transmission pattern, we are just starting to also piece together the topologies of the host populations for bacterial species. And there's this beautiful paper, one of them is this beautiful paper by Gordo et al. In 2023. So the theoretical question that I'm going to focus on tonight and that I wanted to tell you about today is very briefly formed, right? It's very short. How does population structure, be it spatial structure or structure of interaction, shape evolutionary dynamics in general, right? And we're by no means the first to think about this problem or this question. In fact, there's many previous modeling approaches in population genetics, in evolutionary biology and theoretical population biology that try to understand the role of spatial structure in shaping evolutionary dynamics. And this goes back all the way to beautiful work by Maruyama in the 70s and so on. However, usually when people represent spatial structure and want to ask what is the role of population structure in shaping evolution, researchers represent space as distinct number of patches or demes, very symmetrically connected by migration corridors. And I'm showing here on the slide some of these types of models that people usually consider, right? Very symmetric models where you have demes or patches, all connected by migration corridors, or like in the stepping stone model, more like a lattice-based model where you have nodes connected symmetrically to their neighbors. Increasingly, people are starting to study more spatially explicit, yet still in some limit, very symmetric metapopulation models, more at the interface of ecology and evolution. Now, the benefit of some of these approaches is that models are very intuitive, they're very simple, and they're analytically tractable. So it's very easy, well, not easy, let me not say that they're easy, but it's easier if you have a symmetric model to mathematically explore it and understand it, right? And being able, through mathematical exploration, we are able to also get a lot of intuition into why, what is the mechanism that is driving the results in these models. Now, the findings for these class of models is that unless one makes assumptions on differences in selection or periodic extinctions or some kind of assumptions that make the nodes different somehow, spatial structures usually do not change probabilities of fixation of new mutations and just affect times to fixation. So for people who haven't really encountered probabilities and times to fixation, think of them as proxies, evolutionary proxies for let's say the process of evolution in a population, right? So when we mention probabilities of fixation, we usually think about a population that is at some sort of equilibrium. So let's say we're fixed on some kind of wild type mutation, and then we have a new mutant appearing in the population. And we want to ask, right, as the frequency of this new mutant is shaped by its selective advantage, the force of randomness and drift, and all kinds of other evolutionary forces, what is the probability that this new mutant starts to increase in numbers in the population and eventually fix in the population and replace the wild type, thus changing the makeup of the population? So probability of fixation of a new mutant starting from one copy, together with the time to fixation, right? How long it takes for this new mutant to replace the wild type, becomes a very useful mathematical proxy for what happens and how fast evolution can proceed, right? And starting from this kind of concept and these proxies of what happens for one mutant, then we can sort of extrapolate to multiple mutations and build more complicated theories of clonal interference and hitchhiking and selective sweeps and so on, right? All of them have at their core, right, these changes in probabilities and times to fixation. Now, as I mentioned before, again, the benefits of these models is they're intuitive, they're analytically tractable, they're easier to work with. However, the challenge is that their predictions are very hard to use for existing, like the massive amounts of existing data sets of spatial organization that we are just starting to have in population biology. We are starting to see incredibly complex patterns of localization, co-localization of an interaction. And again, remember some of these spatial structures that I've showed you before. The problem is that if I want to study more complex topologies, it's much harder to study. So I have this challenge that I have a theory of evolutionary dynamics in space that is built mostly around these very symmetric models. And it's incredibly hard to link their assumptions and their results to these very complex, heterogeneous data sets of spatial organization. It's very hard to know how to use the models to understand and infer from data. And as we try, right, to represent these data sets, and as we try to think of other representations, spatial representations for these data sets, the challenge really lies in finding, okay, what are the right geometric representations that help me capture their heterogeneity, their complexity, but also, what are the geometric representations that allow for modeling tractability? Because I still want to be able to mathematically model and understand and get intuitive and heuristic understanding as to why particular spaces may accelerate or decelerate evolution, as to what properties of space matter for evolution, right? And something that we kind of settled on very early on is to use this mathematical proxy of networks, right, of graphs. Graphs and networks appear in so many different fields these days and can be a transformative tool for many different types of questions. They can be incredibly versatile mathematical representations, including representations of population structure and spatial structure. And they almost come, they almost represent some of these types of topologies that we have to deal with intuitively, right? They're a natural, intuitive mathematical representation. Even for the types of structures that are not social networks, right? That may be where you're thinking, I don't know how intuitive networks would be to look inside the human body at a much smaller scale of organization. It turns out that networks still can capture these types of ductile properties of different structures, and we can build networks to try to represent these structures as well. So an additional benefit of networks is that they also help us connect our potential results to prior models and results, because a fully connected population, we call it this well-mixed population where every individual can be replaced or interacts with every other individual, can still be represented through a network, and that is the complete graph. An island model where you have the different demes, where everything is connected by to every other deme, can also be represented by a network. The stepping stone model or a lattice model can also be represented by a network that is so-called the K-regular graph, right? The graph where every neighbor has the same number of nodes. when I say nodes here, it's important to sort of think, okay, what do nodes and links mean in this network? So whenever we think of a network, we can think nodes can be either one individual, and this depends very much on the application, right? I'm going to keep this talk very general, very mathematically sort of general, but as we start to think about applications, the details matter. So a node can be either an individual or a subgroup of individuals, a subpopulation, right? That is homogeneous within that subpopulation, right? So a node can also be a DEM if we make certain separation of timescale assumptions. A link between nodes represent the patterns of reproduction and replacement, or the patterns of interaction in the population, right? So that's how we should think of nodes and edges. So then, we've kind of made the problem even more concrete. And the problem becomes, okay, let me assume that I have a population on a network, right? And I have this new mutant, this pink new mutant appearing in a random node on the network. And I'm going to detail in a minute how the mutant, what happens at every time step in this evolutionary process. But I want to track the spread of this mutant through this network. I want to see when and if this mutant fixes, what is its probability of fixation, right? And particularly, the very specific problem becomes, how does the population structure, right, the network structure, affect the spread and accumulation of this new mutant? So to do that, we're going to use the so-called Moran process, a very famous process in evolutionary dynamics, which is a birth-death process. So the rules of the stochastic process are such: at every time step, I select one of the nodes or one of the DEMs for birth with probability proportional to fitness, right? So every node, right? Let's say I have the mutant on the node or the wild type on the node. The mutant has a particular fitness advantage or disadvantage, same with the wild type. So there will be nodes of differential fitness in my network. I select a node proportional to fitness for birth. I select one of its neighbors for death. And this is sort of the crux of the problem becoming a population structure problem compared to a well-mixed problem. If it were a well-mixed population where structure would not matter, then the nodes selected for death in those models would be from the entire population, right? That would make it well-mixed. But here, the pattern of reproduction and replacement is local. So I select a node for death. And then I replace this node selected for death with the offspring of the initial node, right? So I have this Moran process, this birth-death dynamics on the graph that tells us, right, this the patterns of reproduction and replacement that can happen on this topology, right, on this architecture. And what as we start to study this process on different graphs, what we and others have found over the years is that graphs, right, these types of structures with more heterogeneity, start showing differences from the prior symmetrical structures that I've told you before. So as soon as I start putting these mutants on more complex population structures, I'm starting to get so-called graphs that are amplifiers of selection. So what are these graphs? Graphs that are amplifiers of selection are graphs where, compared to a well-mixed population, so that's important to note, I always have a baseline or a null, right? I always, in order to study the effect of population structure, I always compare evolutionary dynamics to what would happen if there were no structure. So I would compare what would happen in a well-mixed population, right? So what we're starting to see is that there are structures where if, let's say, a beneficial mutation appears, these structures almost like help this beneficial mutation. So advantageous mutants are more likely to take over and replace the wild type on these types of structures, right, compared to a well-mixed population. At the same time, for the same type of structure, disadvantageous mutants, so bad mutants, are more constrained. They're less likely to fix compared to a well-mixed. So this is shown in this graph where we have selection strength on the x-axis, fixation probability of these mutants on the y-axis. The black line is the well-mixed. And we see that as soon as we have positive selection, these amplifier graphs, right, do better than the well-mixed. And the amplifier graphs do worse than well-mixed for deleterious mutants, right? Another way to think about it for people who know a little bit more evolution is that these graphs, almost like in this tug of war between the forces of evolution, between selection and drift, almost help the force of natural selection. The balance tilts towards selection and against drift. There are also another category of graphs that is almost the opposite of this, where if we compare to a well-mixed population, good mutants are less likely to spread, right? They're kind of like the population structure makes it harder for them to get a foothold in the population and spread. And the opposite is true for disadvantageous mutants. So in that case, the balance of forces tilts against the force of natural selection. And in the middle, right, where we've said that probabilities of fixation are not changed, are exactly these types of so-called isothermal structures, these types of very symmetric structures that people have studied before, right? So already, right, we're starting to see that as soon as we start introducing a little bit more complexity in the population structure considered, you're starting to move away from these prior results that spatial structure, again, in the absence of other types of assumptions that would force it, spatial structure does not change probabilities of fixation of new mutants. It does not help a mutant in any way to spread, whether it's in a well-mixed population or on a particular type of architecture. So at a very big picture level, again, for people who know a little bit more evolution, so it's not necessary to understand this if you haven't encountered this before, this essentially happens because you have this local heterogeneity in these node reproductive advantages, right? The number of neighbors each node has creates this local heterogeneity. And that essentially at a graph level makes these graphs amplify or suppress selection. And I'll come back and detail a little bit more and what I mean by this. Now, something to point out as we start to work with these networks and try to understand these results is that these networks, networks are amazing. They're so versatile and they're a wonderful mathematical tool to work with. However, they also present significant challenges, and it's important to consider these challenges, especially because I'm going to sweep a lot of things under the rug, like a lot of the details, the mathematical approaches, it can be quite a lot, and you can find all of those in our paper. But for the purpose of the story today and to keep the talk manageable, so many details and so many sort of hard things about networks, I'm just going to sweep under the rug today. But just to give you a sense of some of these challenges and what makes these problems hard. So the first challenge when it comes to trying to understand dynamical processes, right, the evolutionary process or a dynamical process like this on a network, is that these models can be quite difficult to analyze, right? You have these stochastic models, right? These birth-death models that sometimes can be difficult even in the well-mixed limit. And then you have these multidimensional sort of properties through having multiple mutants on these networks. So even, if you have only one mutant or single mutation frameworks, people have had a really hard time studying these networks and their properties. And most prior work has been done in a limit of either incredibly small population size. So here, for example, this is one of my favorite papers from Arne Tralson's group. And you can see he studied networks of n equals 6, right? So again, not quite a symmetric model, but still we're like, these networks that we studied, to be fair, are still quite symmetric, right? You have the cycle, you have the connected graph shown on this picture, but still very small networks that, again, make it very hard to link to more realistic data sets. The second challenge, when it comes to sort of more generally this sort of subfield, right, or these types of approaches, is that in order to make some kind of systematic, rigorous progress and mathematical progress, people have started studying quite artificial network structures. So there's many previous studies that focus on particular graph families, such as the star, right, where you have this node in the center with a lot of nodes around it, the leaves around it, or superstars or funnels or bipartite graphs. And there are benefits to studying particular structures, right? So again, the more symmetric the structure is, the more control I have over it, the easier it is to manipulate, right? And to study and to mathematically understand. However, if we focus on these structures can be very artificial when it comes to thinking about a biological systems that could potentially have these structures. So they don't really help us understand more realistic networks. And also, they make it hard to build a unified view of how network structure in general, right, or general network properties shape evolutionary dynamics, right? What about if I have a structure from a particular data set that has thousands of nodes and that doesn't fit any of these particular family structure studied before, right? So in order to make unified progress, we need to understand how different network families compare with each other, but also be able to study structures that we have not seen before and structures that have high degrees of complexity at scale. The other challenge, as we've been dealing with these networks now for a few years, is that, again, they can be mathematically messy, right? Whenever we think about graphs or networks, most of us are familiar with graphs and networks by now. They're incredibly useful as a mathematical tool. But we also know that nodes have many properties. You have mean degree, variance in degree, mixing patterns, betweenness centrality, diameter, clustering coefficient. So you have these tens of properties that I can quantify, right? All of these properties correlate with each other. It's very hard to change them independently of each other. And so it's very hard to know what property to study and where to look in order to understand what actually affects evolutionary dynamics, right? So if just by looking at a review on all the different network properties that were studied, we can see that there's up to hundreds of different properties at very different sort of scales of organization, either global or local or distance-based features. And it's very hard to know where to even begin. So things can seem very difficult and messy, right? Not only do we want to deal with difficult and messy spatial structures, but as we look at these mathematical representations, they seem difficult and messy themselves. But what I want to tell you about tonight is that we've actually managed to make a lot of progress in our work by computing and formally understanding, writing out two evolutionary properties of these networks. These two evolutionary properties of the networks are two properties that are essentially a combination of some of these network properties that I've showed you here before, right? And we found this right combination of network properties that evolutionarily together matter, right? And these two evolutionary properties are essentially the amplification, what we call the amplification factor and the acceleration factor. And sort of for the rest of the talk, I'm going to give you a little bit of a very brief introduction to both of them and to sort of show you how both of them essentially interplay to shape evolutionary dynamics on a graph more generally. Something else that I'm going to sweep under the rug is that what allows us to do this and to present the results that I'm going to show you are not only mathematical approaches and results, but also the fact that we are able to build these edge swap algorithms that allow us to change network properties independently of each other, right? That allow us to understand specific roles of network properties independent of other properties. Because remember, I've told you before that a challenge has been that I cannot understand how variance in degree of a network, for example, affects fixation probability, because as I vary variance in degree, the mean degree also matters. So we've been able to create these edge swap algorithms that allow us to, for example, keep mean degree constant and vary variance in degree. So let me start again very briefly with the amplification factor of a network. So remember that I told you that graphs can be isothermal, so they don't change fixation probabilities. They can amplify selection, they can suppress selection. Well, the amplification factor of a network essentially quantifies this property, right? By how much, right, is a graph an amplifier or a suppressor? So it's a quantification of this property of amplification or suppression. So again, if I plot this amplification parameter, right, on the x-axis, I plot probability of fixation on the y-axis here. The well-mixed model, right, I can compute the fixation probability, and the well-mixed population or structure has amplification 1, right? So this is exactly that isothermal graph, like it doesn't, that's my null, right? Then what we're able to see and get approximations for is that indeed I can study this amplification property, right, across network families for networks of many different thousands of nodes. So what I'm showing you here, the line is an analytic approximation that I'm not going to go over tonight how we got this analytic approximation, but you can find it in our paper. The dots are simulations to show how well our analytics matches generally the simulations, right? And what you can see is that there exist, right, all these different network families that have amplification larger than 1. These are the amplifier graphs, right? And we can sort of quantify by how much they amplify. Then we can see graphs that are suppressors of selection, right? They have amplification parameter lower than 1. And indeed, the probability of fixation here, we use a beneficial mutation. The probability of fixation is under that of the well-mixed. And this also allows us to essentially study the probability of fixation for any new network that you give us, because I can compute analytically or using simulation this amplification parameter, and then I can see, right, what the probability of fixation is. I can have a predicted probability of fixation. Now, the benefit of this amplification parameter is that it also helps us understand the role of all these other network properties that we're more familiar with, like moments of the degree distribution. So for example, if we look at mean degree, so here, all of the dots of the same color are networks of the same mean degree, right? So if I look at mean degree, I can see that as I increase mean degree, so as I make the network more and more connected, as these are represented by the darker blue, I approach a well-mixed population, right? Which is intuitive, right? As I decrease connectivity and I make my network more complex and sparser, I move away from the results of a well-mixed population where everything is connected. At the same time, if I study the variance of the degree distribution, right, here, again, all the nodes of the same color have the same mean degree. So I control for mean degree and I can just vary the variance in degree. And I can see that as I increase this complexity of a network, so as I increase the variance in degree distribution, I move away from the results in the well-mixed approximations or lattice-based approximations. Now, something that's very interesting that I wanted to point out is that, you know, mean degree and variance in degree, these very powerful properties of a network, are not the only properties that go in the amplification factor. Here in this plot, right, I'm showing you again probability of fixation, variance in degree or standard deviation in degree on the x-axis, right? All of the networks that I'm showing here, a dot represents, corresponds to one network. All of the networks that I'm showing you here have the exact same mean degree. So for any color here, mean degree does not change. All of the nodes of the same color, for example, let's look at the dark green nodes, as you can see, have the exact same variance in degree. So the dark green graphs or the dark green dots here all have the same mean degree and variance in degree. Yet it is very easy to see that we have this large range of probabilities of fixation, which shows us that mean degree variance in degree is not sufficient. And indeed, this happens because properties of edges or the mixing pattern, the variability of degree connection, also plays an essential role in shaping this amplification parameter. So this mixing pattern essentially tells us how often do I connect nodes of low degree with nodes of high degree, right? What's the correlation in degree for neighbors? And indeed, right, for the exact same networks, I can see that correlation in degree does affect my alpha, my amplification factor of the network. And as soon as I control for correlation in degree, right, and I put alpha now, this amplification factor on the x-axis, now my probability of fixation beautifully is explained by the amplification factor, right? So if I didn't have this amplification factor, I could, you know, just by doing plots like this, I could wrongly assume that I can explain a lot of what I'm seeing in trends in probability of fixation. But this plot tells me that, hey, no, there are other things at play and I have to correlate, I have to account for that. And indeed, alpha beautifully accounts for that. And this is a benefit, especially for the students in the room. I want to say this is a major benefit of mathematical approaches, right? Because by writing out what alpha is mathematically, and also having computer algorithms that allow us to rewire the network, we can actually track, hey, how does amplification factor change under one rewiring step, right? And I can see this delta, right? Like what happens under one rewiring step as I rewire the network. And this brings me a whole ton of intuition, right? And this shows me mathematically, hey, amplification factor increases when there are more edges connecting nodes of low degree to nodes of high degree. So this allows me to understand why networks amplify or suppress as well. Mathematics can be very powerful in bringing this intuition in terms of mechanism. Now, again, having these types of results allows me to not only understand network properties, but allows me to start designing, right, all kinds of different structures that push the limits of amplification and suppression, and also sort of try to understand, okay, can I cover, right? How would I design a structure with a given amplification or suppression? And this is what we kind of try to do here, to try to see, can we cover the entire space between the minima and the maxima amplification that we can find. Okay, if there's no questions, I'm going to move to the acceleration factor of the network. So I've told you about the amplification factor. That was centered around this idea of how does spatial structure change probabilities of fixation of mutants in a population. Now, the acceleration factor tells us something about how long that takes, right? So let's say I have my mutant fixing in the population, but it can take a very long time for it to fix, or it can take a very short time. And even intuitively, we can think, well, rates of evolution are probably affected by whether something takes a long time or a very short time. Now, what people have seen before, again, on very small graphs, these were all graphs of size n equals 10. So again, very small population sizes. Remember that all the graphs I've showed you before, population size doesn't really matter. We can do this for networks of thousands or tens of thousands of nodes. What previous papers have seen before is that there are these trade-offs between fixation probability and fixation time. And what do I mean by these trade-offs? I mean that, I can design a graph to increase, right, the probability that a beneficial mutation spreads through a population. But that also comes at a cost of the fact that this will also probably take a long time. So as I increase probability of fixation, I usually also increase fixation time, right? So we became very interested in figuring out if there would be a way to decrease time to fixation for the same probability of fixation, right? Can I design a graph where, given a probability of fixation that I want, right? I'm willing to I want to control for that. Can I redesign the graph so that the time to fixation, right, can also be manipulated, and I can decrease it, right? Because then I can get rid of this trade-off a little bit. And we were very inspired actually by two very different types of papers, one a connectomics paper and one a really old paper from the 1960s that essentially gave us this idea to look at network motifs. So network motifs are these types of node clusterings that are of size 3 and more, right? So what do I mean by that? If I look at lower order motifs, right, are essentially nodes and links, edges, right? And you have these motifs of degree 1, right? The nodes, degree 2, the edges, and we've mentioned how properties of nodes are mean degree, variance in degree. Properties of edges are mixing pattern, right? So this correlation in degree between nodes. Now, these higher order motifs are degree 3 and above. So at first, for example, for degree 3, I can have triangles. So these cycles, these connections of 3 nodes. And then the opposite of that is wedges. So this type of sort of V-shaped pattern. And then as you go down in terms of, up in terms of degree, you start to see more and more motifs for a degree larger than 4. So again, we played with a lot of analytical approaches, and we designed algorithms that allow us to play with these cycles and motifs and keep the lower degree properties constant, right? Keep properties of nodes and edges constant. And we realized that, for example, as we go to these types of cycles, just like the triangles, right? And we vary them, they don't change probabilities of fixation, right? So here I'm showing you different networks where the colors are different mean degree. all of these networks essentially are the same for the same color in terms of lower degree properties, right? And I'm plotting the fraction of triangles on the x-axis, the probability of fixation on the y-axis. And we can see that if I just change the number of triangles in a network, again, without affecting mean or variance in degree, nothing happens to the probability of fixation, right? But as soon as I change the fraction of triangles, right, even though the probability of fixation is not changed, the time to fixation is drastically increased, right? So these higher order motifs do not change probabilities, but can change time to fixation. So this allows us to study, right, independent of amplification properties, this allows us to study these acceleration properties of the network and be able to analytically write out these acceleration factors, right? By how much does the network speed up the spread of a mutant compared to what happens on a well-mixed population in terms of other properties of the network. And in the paper that I mentioned here, we also sort of talk more about the why. Like, again, out of the analytics comes sort of this intuitive understanding. And it turns out that, you know, triangles or these cycles, essentially, these cycle connection between nodes, reduce the number of edges where mutant and wild type meet.
Preguntar algo, Gustavo?
Sure. Yeah. 1 second to finish the sentence, and I'll give you time for a question. So again, by these cycles essentially end up reducing, right, these interfaces, these meeting points between mutant and wild type, and that is what affects evolutionary dynamics in the long term. So again, mathematical intuition is very important for understanding why. Why do we see the results that we're seeing? Yes, go ahead, Alberto.
Using these strategies of examining networks, you arrived at certain predictions. I guess you have tried to test experimentally some of these predictions.
So the predictions that we're seeing, right? And the way I think about some of these, I think this is a great question and it's very important. The way I think about these mathematical models is that one can use them to get sort of this unified understanding, right? To get us almost intuition into what types of structures should I expect to amplify or suppress things. In order to validate or to test, right, there's two different avenues, right? One can use mathematical models together with networks that are inferred from real data and then make predictions and see if the predictions match sort of the rates of evolution that we see in real data. And we're doing some of that work. And I'm going to tell you a little bit about one small example of that later in the talk. The other way to validate or test these predictions is to actually design some of these structures in the lab and then see whether they amplify or suppress, or what other, and if they don't, what other, what are we not accounting for in the mathematical models? And indeed, I have a PhD student together with an experimental collaborator where we're validating some of these results in the lab with bacteria. But those are very preliminary results, and I'm not going to talk about today.
Great. Thank you very much.
Mm-hmm. Any other questions?
I don't see your hand up.
Okay, so let's move on. So, you know, I know we're running out of time. So in the last few minutes, I wanted to say, you know, as we start to put these together, right, this opens the door to all kinds of explorations that are not in this very strict limit of one mutant, right? So I've told you about these models where, if you think about it, they're incredibly restrictive, right? Hey, she's studying the probability of fixation of one mutant on this fixed network, right? There's a lot of assumptions here. But nonetheless, these types of mathematical models allow us to start making progress on a lot of other more complicated types of problems, right? That are not as restrictive, and they provide a very sort of solid baseline in which to help with more complicated problems. For example, you know, if we want to move away from this assumption of a single mutant spreading through this network, we find that these results and these two evolutionary parameters, amplification and acceleration, explain also what happens in a regime of recurrent mutation when you have clonal interference, for example, between mutations. So as different mutants arrive in the population over time, right? So I have, for example, here mutant A, then on the background of A, I have another mutant and another time point, right? Previously, people have been very interested in the role of space, for example, in shaping clonal interference or sort of clonal war between these different mutants. And the consensus in the field is that as you introduce space in a population and spatial structure, because all of these symmetric spatial structures don't change probabilities of fixation, but change the time that it takes mutants to fix. So they increase the time that it takes mutants to fix. That means that these mutants are sticking around for longer. And so you should see, right, more clonal interference, right? More as good clones come into the population, they fight with each other more. They interfere in their spread with each other more. And this leads to less mutational utilization and lower rates of evolution, right? So even though maybe we have good beneficial mutations coming into the population, spatial populations with spatial structure usually show lower rates of evolution because of these this process of clonal interference and lower mutational utilization. And you can also see it in some of our work looking at all these different types of spatial structures. So for example, here, we're just playing with these number of cycles in the network. So for the 3 networks that I'm showing you here, all of the nodes have the exact same mean degree 8, right? Because we wanted to particularly compare to a lattice where previous results have been derived. So every node here has 8 neighbors, but we're just playing with the number of triangles or cycles of higher dimensions in the network. And this leads them to look very differently. And indeed, as you make these Mueller plots looking at competition of different mutants in the population, you can see that just by changing number of triangles, you have very different evolutionary patterns that emerge. And we can quantify that, and we can sort of do it for spatial graphs too, or sort of more clustered, less clustered. But our results allow us to essentially quantify these rates of evolution and study the role of amplification and acceleration independently. And then we are able to show that just by understanding the limit of one mutant, amplification and acceleration, again, right? Because that's what was important for one mutant. Exactly those 2 parameters also help me understand multiple mutants, because I can show that the results only need these 2 parameters that I derived for the case of one mutant. And the way we show it is to show that the equations, right, that govern the evolutionary dynamics in the case of space are just like the equations that govern the evolutionary dynamics in the case of a well-mixed populations, but with the right stretches to the parameters. And these right stretches are given by the amplification and acceleration factors. And again, by understanding the amplification and the acceleration factor, I can also start putting a lot of different network families together and new networks that maybe don't belong to a family or that I'm inferring from data. And we're starting now that we have some of these theoretical predictions, we're starting to link models and data. So we're starting to look at different type of data sets where we have inferred the spatial structure. For example, in ductal carcinomas, we see as expected that the rate of evolution is higher than, let's say, if the same tumor were modeled on a network, on a lattice, or a more symmetric graph. Now, again, this is not necessarily validation for these results. That's just a small initial hint that maybe there is something here in terms of these mathematical results being very useful for a wider class of problems with more specifications for the system of interest, to try to understand the role of spatial structure. So we're starting to get hints both from our network inferences, but also from some of the lab work that I'm not going to tell you about today, that these theoretical results do match the observations. So just to give an overview and to wrap up, using network representations, we can show in theoretical models that we can capture evolutionary dynamics that simply is impossible with lattice-based or Dean-based regular structures that we usually default to when we try to understand the role of space in evolution. And the other thing I want to say is that we may think that these networks are messy and hard to work with and hard to study. But what we found in our work is that there's actually a lot of unified understanding that can be obtained and that can be extended to a lot more complicated models. But we were very surprised when we saw that clonal interference. We also have results on stochastic tunneling, crossing fitness landscapes, so epistasis, results of a lot more complicated models that I didn't have time to tell you about today. We were incredibly surprised to see that the amplification and acceleration factors, just these two parameters that we discovered, can help us understand even a lot more complicated evolutionary processes. And so, these are still very analytically hard problems, but they can, I believe, provide a lot of unified understanding. And at a big picture level, we think a lot in my group about simple, parsimonious enough models. And I do believe it is important to understand, right, for forming null expectations about what we should observe in experimental or observational data. It is very important to understand the role of more complex spatial structure or the role of more complex topographies, right? Not everything sits on a plane in 2D, right? What about the topographical structures that we see in the world? It's very important to consider, even with mathematical models or with experiments, to consider how complex topologies can actually impact evolution, because it's important, right? When we're trying to understand our systems, it's important to develop an intuition. Okay, when is it enough to just not even care about space? When is it enough to just assume, hey, this is a well-mixed approximation, which a lot of, including models of cancer, or a lot of models do. If I want to understand space, when is it enough, right? When is it simple enough to put it on a lattice or on a two-patch system or on a connected DEM type of spatial structure, right? Maybe that's enough. And maybe that's enough for the model, right? But we shouldn't necessarily default to using lattices as spatial approximations if our data potentially has a lot of complexity in its structure, because then results from well-mixed approximations, results from all of these isothermal type of structures like lattices or very symmetric structures simply might not apply, right? So that's sort of the kind of take-home message that I wanted to share today. So many people in my lab contributed to these results, but especially my student who's taking the picture here, YP. I'm very grateful to my students. We're always recruiting at Carnegie Mellon. I'm especially recruiting postdocs and graduate students. So if you know of good students that might be interested in some of these topics, please send them to me. And I thank you so much for your attention, and I'll take any questions.
Thank you, Ana. So, por favor, levanten la mano y abran el micrófono para las preguntas, los que estamos aquí en Zoom. Los que están en YouTube, por favor, escriban sus preguntas en el chat y aquí las leemos. Diego.
Hi, thank you very much again for the talk. It was very interesting. I have two questions. The first question is, in other domains of biological networks, such as gene regulatory networks, it is often argued that the gene regulatory networks follow a power law in their degree distribution, and they say that this might give some properties of robustness to the network, because if mutation appears in a lower degree node that constitute the most part of the network, the effects of the mutation are not going to affect the whole structure of the system. I want to ask if there's anything similar in these population structures that might explain some evolutionary constraints in the I don't know, in the structures that are preferred in nature.
Yeah, I think that's a great question, right? And I think your question, again, goes back to this, let's say, amplification parameter of a network. So what are the properties of the nodes, right, that control for the amplification parameter? And you mentioned, let's say, networks that have a lot of low degree nodes, right? So there's two things when it comes to a network, right? So for all of the networks that we studied here, it's important to note, and I'm sorry I didn't mention it, that all of the simulations were essentially averaged over, let's assume the new mutant starts in a random node of the network. So I cannot pick and choose my node as a starting point. A mutant just appears in the population and I have no control over where it appears, right? So a lot of these global properties of the network, like the amplification and the acceleration factor, are given by the fact that a mutant can start anywhere, right? And I'm averaging over all these different starting points. And so it really is a global property of the network. Now there is separate work where if you control for the node, for the starting node, it turns out that, of course, that makes a difference for probabilities of fixation. So if I pick my starting node, I actually have what's it, inverse relation. So the lower the mean degree, the lower the degree of the starting node, the higher the probability of fixation of the mutant. And I know it's not intuitive, but it goes back to the comment that I made that actually low degree nodes are better to be in because they're less likely to be replaced. If I'm in a hub, right, and I have nodes, I'm surrounded by nodes, and I have a lot of nodes around me that can potentially replace me, right, in this birth-death process, it's actually not good for the hub, right? So it's not beneficial for me to be in a hub. It's really the low degree nodes that it's good to be in. So if I control for the node, right, then the results start changing a little bit. So it's a different, slightly a different flavor of result, but that's a great question.
Very interesting. Thank you very much. I have another question about the acceleration factor. You mentioned that this factor is mainly associated with the proportion of triangles in the network. And there's a property of networks that is the clustering coefficient distribution that also approximates the proportion of triangles that are formed in the network. So I want to ask if you have a look how this acceleration factor is related to the shape of the uh, clustering coefficient distribution.
I have not looked at that, but I just wrote it down. I think that I'm gonna look into it. If it's correlated with triangles, right? So we were able to explain intuitively why we see the results that we see evolutionarily, because these cycles have their particular properties. So as long as the clustering coefficient distribution is correlated with these sort of the appearance of these cycles, maybe that's another way to explain some of the results. Thank you for that.
Thank you.
I'm going to read a question from YouTube. Interesting. Thank you, Dr. Carja. Will this help how we design artificial neural networks architecture?
I'm going to say yes. We're working on that very preliminarily. We're working on essentially, we have so many different kinds of problems around that. Like we can show, for example, that we can design networks that help us cross fitness valleys more efficiently, right? Faster and with higher probability in fitness space. And that applies to questions in cultural evolution, for example. So if you need to have communication between different agents, usually when there's an innovation in communication, so let's say I have an innovation in the sender, but the receiver doesn't really have the innovation yet, then you have to cross a fitness valley. And so some of our networks we can design to make that more efficient and with higher probability. And so there's a lot of different agent type problems that we're playing with. We haven't necessarily started playing with neural networks, but if you're interested, come talk to me. We're recruiting.
Beto, ¿quieres hacer tu pregunta?
Sí,
Adelante.
Thank you very much. It was a great talk. Have you thought, or is it known if there is a relation between the spatial topography and criticality in networks?
Can you tell me what you mean by criticality?
Well, it's this general behavior of biological networks that tend to a critical slope, which means that they are very capable of handling small changes without deriving from their basal behavior.
Mm-hmm. I see. So, and I think, yeah, I see people have studied that, let's say in protein-protein interaction networks and all of these other, and it links a little bit to the robustness comment from before. I haven't really looked at that. I think what your question makes me think of is this other question from yesterday where a student asked, well, if you look at a lot of different biological networks, right, and you try to put them or cluster them in one of your plots in terms of amplification and acceleration, like how do you link to these, like where do they cluster, where do they all sit, and how do they link to these other properties? So I think your question is very similar to that, right? Like biological questions, right? how much amplification and acceleration do they show as we gather all kinds of different biological networks, and how are these evolutionary properties that I'm mentioning today linked to these other properties around robustness and criticality and so on. So I think these are very good questions. We just haven't started exploring them, but these are definitely sort of next steps that we're going to take. I think it would be beautiful to look at, try to gather many different networks types and try to sort of get more intuition. Where are we in the space of parameters? Yeah, thank you for that.
Thank you.
No, ¿otra pregunta? I don't see any more hands up. So, Oana, thank you very much again for your very nice talk and very nice results. And I'm sure it's a big future studying, applying your knowledge into biological questions will be great in the future.
And thank you so much for this opportunity. And maybe I get, 10 years from now, I get to tell you about all of the results from the questions you asked today.
Thank you very much.
Thank you. Okay.
Thank you so much.
Thank you.
I'm trying to stop sharing and
there was another comment, so probably you can read it in the chat now when you close your presentation, if you manage to close it.
Yeah, I think because I can't don't have my mouse, I don't know how to exit. Stop share. Okay, maybe I can. Tab. Oh man. Will I